I guess first thing I have to say is Raphael's a very tough act to follow. I don't have any videos anything nearly as good as that. Maybe more importantly, first, Seb, congratulations on your honor. Very well deserved. Also, I think you did a fantastic job with the symposium. I appreciate greatly being included in this group. Tremendous talks. And also, I've known Seb, I think, about 25 years. We met at a Gordon conference back when you were still working with Sam, maybe around 2000. And we've interacted almost those whole 25 years, I think. So it's been a pleasure collaborating, visiting each other, seeing each other at conferences, working on biomacromolecules, all these different things. So you've been a great colleague, collaborator, also a great friend. So today, back off a little bit. I'm too loud. There we go. So today what I'm going to focus my talk on is dehydroalanine, specifically polydehydroalanine. So the dehydroalanine residue is here. This was interesting to our lab for a couple reasons. One was the chain conformation, if you have a peptide made up only of dehydroalanine residues. This is a crystal structure here from Claudio Toniolo's group. They crystallize very short oligomers of dehydroalanine. What's really striking is this peptide is completely flat. So if you think of polypeptides, you think alpha helices, beta sheets. This is completely different. All of these residues are in a plane. That was really weird. Also, it has intra-residue hydrogen bonding. So this is the nitrogen, the alpha carbon, the carbonyl. There's a hydrogen bond within the same amino acid residue. Very strange material. We were really interested in how is this going to behave in a material. If we want to do supramolecular chemistry, assemble these. Our group and others have done a lot of work in the past assembling polypeptides that have alpha helical segments, beta sheet containing segments. What will that do? Can we get layered type assemblies? So we were very interested in the structure. And then also, of course, dehydroalanine is very well known in terms of reactivity. It is a residue that does occur naturally in various biosynthetic pathways. Typically, there's either a serine or a cysteine residue that gets dehydrated, usually enzymatically. You make this dehydroalanine, which is very electrophilic, being an alpha-beta unsaturated alkene. Usually something, a nucleophile like a thiol can react with this and make a stable bond. This occurs in the formation of things like cyclic antibiotics, like these lantibiotics. And so it's a way to have an electrophilic functionality on a polypeptide. Normally, polypeptides have nucleophilic functionality, so we can switch things up. And so we thought this is also a great way to potentially functionalize polypeptides. One thing that's worth pointing out is, at least in the biosynthesis, you will start with something like an L-amino acid. Dehydroalanine has no chirality. When you react it with the nucleophile, this is actually incorrect. It was from this review article. When you do modify this, this is typically racemic. So you are going to make more disorder in your stereochemistry, heterochiral materials, once you do this modification. So they have a potentially unique conformation and also very interesting functionality. The functionality has also been looked at quite extensively in the chemical biology community. Ben Davis's lab has done a lot of work in this area and also has been followed up by others. This residue can be installed usually from a precursor such as cysteine. It can be installed site-specifically into proteins, and then if you come in with a nucleophile, you can modify that very selectively, and recently people have also done radical additions to DHA residues. So it's a very nice way to have a fairly unique reactivity in a protein where you can site-specifically attach various functional groups. One thing we really liked about this is all of this chemistry, of course, works very well under very mild aqueous conditions that can mimic what you find in biological systems. So how do we make polydehydroalanine? Well, from a synthetic procedure, as we've heard earlier today, lots of synthetic polypeptides are made by N-carboxy anhydrides. That's the obvious choice. In fact, Sakakibara published a number of papers in the late 1950s and early 1960s where he made dihydroalanine NCA. That works very well. It's actually a quite stable NCA. It's a little more electron rich than other ones, making it a little less reactive. However, when he tried to polymerize this with a variety of initiators, you really don't get polydihydroalanine out. You get something, I think what he was best able to describe as something that looks more like that, where you really have very little alkene content in the polymer. The reason for this is when you add an amine onto the NCA, first of all, the NCA is a little less reactive, but the amine you get on the chain end is an enamine. This is a lot less reactive than a normal primary amine. It can also rearrange, it can tautomerize. There's lots of things that go on, but it really doesn't polymerize well. We had done a lot of work ourselves trying a variety of different initiators and confirmed Sakigahara's results. You really can't get polymer that has very much alkene content at all in it. I should say that there is a whole different community that does radical polymerization of dihydroalanine NCA to make these hydrocarbon backbone polymers. That's a totally different story. We're not going to talk about that. So the NCA route doesn't really work. So how do we do this? We need an alternative route. What we decided to do was do similar to what a lot of people in the chemical biology community and also nature does, is start with a precursor and then convert it into dihydroalanine. The challenge with polymers is we need a precursor that's going to be soluble so we can control chain lengths, make block copolymers, and also do efficient functionalization. We focused on cysteine-based precursors. We essentially added a tert-butoxy bromoacetate to this to make a tert-butyl carboxymethylcysteine. These types of amino acids can be easily converted to NCAs and then polymerized. A lot of these have been around for quite a while. One problem in this area, though, is with many side alkyl groups here, such as benzyl or methyl, these polymers always form beta sheets. So the methyl carboxymethylcysteine, benzyl carboxymethylcysteine form beta sheets, the polymers precipitate. It's very hard to control chain length, very hard to make things like block copolymers. We had some experience with glycopeptides based on cysteine in the past, where if you have a very bulky group out here, you could prevent beta sheet formation and instead drive these into more of a helical structure. There was also some work done in Japan in the 70s with a similar result. So all we did in this case is we put in this tert-butyl group with the idea that hopefully that would prevent beta sheet formation. And it actually worked quite well. We can make this NCA using fairly standard methodology and then polymerize it very well. We make this tert-butyl carboxymethylcysteine polymer. It's alpha helical, very soluble. We can control the length, control compositions, all these other things. The other thing that's nice is, of course, the tert-butyl group is quite easy to remove, to deprotect, and we use this to convert to polydihydroalanine. So with this precursor in hand, we try to convert this directly to poly-DHA, but it's too hydrophobic, doesn't work very well. But what we do first is we just deprotect this into carboxymethylcysteine polymer. This is charged, very water soluble. The charges break up any beta sheet conformation. And then this is very easy to work with. So we really are not inventing anything new in converting this to DHA. It's well known that if you take a molecule like this, an alkylated cysteine, and you alkylate it to make a sulfonium, these can eliminate very readily. You pull off a proton, the electrons push up, kick out that leaving group, and you get dihydroalanine. This works extremely well. This is a very fast reaction. As soon as we add the alkylating reagent, we never see this polymer or these residues. We just get polydihydroalanine. We use slight excess of alkylating agent. It's quantitative functionalization. So this works as a very nice way to make these dihydroalanine polymers where we have all of the alkenes. Another thing we were very interested in was also using oxidation to drive this transformation. And this is known in individual residues, small peptides. You can oxidize the sulfur to a sulfoxide, and then by heating this, you can eliminate this species and get polydihydroalanine. The best we ever got was maybe about 10% conversion. Believe me, we tried lots of different ways, but this just does not seem to be very efficient. So we're primarily going to focus on this route up here. We're going to make this precursor polymer, deprotect it, alkylate it, and then we get polydehydroalanine. So we really did make this polymer. It's not soluble in a lot of things, but it does disperse quite well in DMSO and hexafluoroisopropanol. Here's a GPC and HFIP. If we look at the proton NMR, we see alkene residences, which is great. We have this polymer, and we also have carbon residences that are consistent with the structure. Note that we do have multiple carbon residences, and I'll come back to that in just one second. So this looks like it works quite well. We did check to see if there was any sign of chain cleavage during this process, but we don't see any. We can incorporate these segments into statistical and block copolypeptides quite easily just by copolymerization. And we were able to, at least to this point, we can make fairly high molecular weight polydehydroalanine. We've gone about DP250, but I don't really see much of an upper limit here. The precursor is very soluble. So the first question is this really flat? And just as a brief refresher here, most of you probably know this, alpha helix, 310 helix, 2,5 helix is this flat conformation. That's the nomenclature that Toniolo gave to this. And then the beta sheet. The 2,5 helix is quite interesting. If you look at the rise per residue or the pitch, it's really the most extended chain conformation. It's more extended than the beta sheet. You're essentially just stretching that chain out completely flat. And it's also the only one that has intra-chain hydrogen bonding. So Toniolo's lab is really the pioneer in this area. They made a lot of different peptides that contain this structure. And they also identified a number of signature infrared bands that are characteristic of this. So we looked at our sample. These are just FTIR of our poly-DHA in the solid state, in the NH region, and also the amide region. And what we see is these green ones are consistent with the 2,5 helix, the flat structure. The most characteristic one is this amide 2 band that's very low, around 1490 reciprocal centimeters, that you really only see that with the 2,5 helix. Another key one is the NH stretching band. This is really not shifted much because the hydrogen bonding is very weak, the intra-chain hydrogen bonding. So it looks like there is some of this 2,5 helix there. However, obviously, there's a lot of other stuff there as well that is not consistent with the 2,5 helix. There is computational work that says the 310 should also be stable, the 310 helix, but none of these bands align up with those. What these do show is this is indicated of inter-residue hydrogen bonding. You can actually get a stronger shift, and we get also other bands that are consistent with that as well. So it looks like there may be 2,5 helix in there, but there's also some other chain conformation, and this is also consistent with these multiple bands we see in the carbon NMR. So this was a little messy. We sought out some help here. I got in touch with my colleague Ken Houck at chemistry at UCLA. He connected Alex Bell, who was a super talented undergrad in my lab, with Declan Evans and his group, and they started doing some computational studies. The first of these was just to analyze the stability of conformations as a function of chain length, and they did this for both the 310 helix and the 2,5 helix, since these were both proposed structures. They got this very interesting correlation with chain length. What they found is if your number of residues is less than 12, you should get a 2,5 helix, the flat structure. And this is exactly what Toniolo sees in his crystal structures. However, if you get above 12 residues, because of increased dispersion interactions and also stronger hydrogen bonding, you should get a 310 helix. So this was interesting, and we would expect to get this 310, but the important thing to take away from this is they only compared these two structures. Our data, experimental data, did not agree with the 310 at all. So Ken is quite smart. He got Declan and Alex to also use this search algorithm CREST that essentially looks for any other possible conformations that might be stable. And they found this. They call it the hybrid coil. I'm not sure if that's super imaginative, but it kind of describes things. But it's a very loosely wound coil that has flat segments in it. So essentially you have a flat strand and a kink, a flat strand and a kink, and it kind of curls around in this very wide coil. The important thing is This has elements that look a lot like the 2,5 helix, but it also has inter-residue hydrogen bonding to stabilize the structure. And at least from these calculations, it's more stable than either the 2,5 or the 3,10 helix. So it looks like this sort of hybrid structure where we do have some flat segments, similar to that crystal structure I showed at the beginning, are present, but we're also getting interchain hydrogen bonding as well. So this is the thing that shocked me the most is this is somewhat consistent with our data where it looks like we have two different types of conformations in these residues. So this is computational. We're still working on experimental data to dig into this further. That may take a while. We're trying to crystallize these chains, but we do get some idea of what the structure. It's definitely not an alpha helix or a beta sheet. So what to do with these? Well, the first thing we wanted to look at was reactivity, and this is some work we published a little while ago when we first recorded the polymer. These were just proof of concept experiments. If we take polydehydroalanine, we usually dissolve this in a DMSO water mixture. It's not soluble in pure water. You can just add a thiol, slightly elevated pH. pH 7 will also work, it's just slower. You can modify these quite easily, and this is very consistent with everything else that's in the literature. These were just model molecules we added onto these. You can make glycopolymers with these as well. The functionalization works very well, and it also works under quite mild conditions. So we started thinking, well, okay, if we can functionalize it, how do we functionalize it? What should we make? So the first thing we looked at, we're trying to make coacervate formers. So our group had reported a few years earlier these polymers. These are optically pure polymers made on a homocysteine backbone where we essentially added amino acids onto the end of the side chain. So the important thing about these is we have these amino acids which are all cationic at physiological pH. And we have hydrophobic R groups, so we have charge and hydrophobicity, giving us some frustration in terms of solubility. These polymers are completely alpha helical, but if you mix them with multivalent anions, they form coacervates. So we said, what happens if we take something like this, add an amino acid with the thiol, add it to dehydroalanine, would that also form coacervates? How would it also change things? So this chemistry is pretty simple to do once you have the polydehydroalanine, so we thought we would do this to compare the properties between these two different types of molecules. So as I mentioned, the chemistry is fairly straightforward. All of the work I'm showing you from here on is done by a really talented grad student, Casey Morrison, who's going to graduate pretty soon. All we have to do mainly is the small molecule chemistry. So we just take a Bach amino acid, add cysteine, reduce it, deprotect it, and we just get an amino acid. The N-terminus is not protected. Free amine is no problem. And we have a free thiol on the carboxylate side. Once we have that, we're in business. And we did this with a number of different hydrophobic amino acids. We just take this, mix it with polydehydroalanine, and we're done. Dialyze it, and we have our completely functionalized polymer. All of these are water soluble as hydrochloride salts. The nice thing here is we were able to include also things like methionine and proline, which were a little tricky with the other method. So the one nice feature about this is synthetically, it's pretty straightforward to make these molecules. But the question is, what would the properties of these be? And the important thing, again, to remember is when we do this thiol addition, these are going to be essentially racemic here. So it turns out these also form coacervates. These are just two examples here. The valine, leucine, and methionine polymers all form coacervates. The alanine and proline do not. We mix these with tripolyphosphate, a multivalent anion. We get droplets that form. A lot of this is very similar to those helical polymers we saw before. The main difference is these tend to be a lot more stable against salt. The ones we had worked with before would dissolve at salt concentrations above maybe 300 millimolar, and these ones are much stable to higher molarity concentrations. We also like this methionine polymer because we can also use it for switching properties. So what I'm talking about here is we have thioether groups both on the cysteine group and also on this thioether group on the methionine residue. These are quite hydrophobic, but we can do mild oxidation with your sort of choice of reagent. We used a couple different ones here. We can oxidize these sulfoxides and make very polar polymers. And what this allows us to do is to reversibly turn on and turn off coacervation, similar to active control that's seen in coacervates that occur in cells. So we make a coacervate with tripolyphosphate. This is at pH 7. Add an oxidizing agent. Again, you can use different ones. It doesn't matter. We oxidize the sulfoxide. This more polar polymer does not form a coacervate at room temperature. Add a thiol, reduce the sulfoxides back to sulfides. We get a coacervate again. So we have a material that can undergo coacervation or dissolution under conditions that are physiologically relevant, and also using reagents that can be found in biological systems, reactive oxygen species or thiols, as may be found in cells. So I'm not going to talk much more about these today, but we have very nice, very uniform, these are all homopolymers, very uniform coacervate formers where we can readily tune their properties. What I want to continue talking about is really more structure. So we're very curious in terms of how these polydehydroalanine hydrophobic segments would affect self-assemblies. We had done a lot of work on block copolymers of amino acids with alpha helical and beta sheet hydrophobic segments, and we learned how those affect assembly. What's going to happen with these? The other thing that we have is these segments, in addition to being hydrophobic, are very stimuli responsive. So these can react very readily with thiols. We go from a hydrophobic molecule, if we use a hydrophilic thiol, we can make something that's water soluble. So we can go from hydrophobic to hydrophilic. Likewise, if we use as our hydrophilic block polymethionine sulfoxide, very water soluble, very biocompatible and degradable polymer, this is also reacted with thiols. This is a disordered polymer. If I treat it with a thiol, physiological pH, I reduce the sulfoxide down to sulfide, just as I showed you before, and I go from a disordered chain into an alpha helix. So we thought this was quite interesting because we could essentially have invertible amphiphils with one stimulus. We're both changing the polarity of both segments. This might be dying. Maybe I'll go to a different laser pointer. There we go. So we have a disordered hydrophilic segment going to an ordered hydrophobic segment, and some kind of structured hydrophobic segment going to a disordered hydrophilic segment. So we thought, well, this would be a nice way to really control, have these responsive materials that essentially could change shape to very mild stimuli. And the goal eventually is these potentially could be useful because these could occur under biologically relevant conditions. So we can make various types of self-assemblies. Where do we start? Well, we chose to start with vesicles. Why vesicles? Well, there's some inspiration. Seb and I go way back. This is a visit that's European date notation, so I think that's just almost 20 years ago. At that time, I was visiting Seb in LCPO. Maybe in the second time I was visiting him. We were both doing a lot of work with polymer vesicles at that time. We were discussing polymer vesicles. If you look, I don't look very happy. I'm not sure why, but Seb actually looks quite happy. He's very excited, very happy. And I think I know why, is he had just published this fantastic paper in JACS where he had done vesicle inversion. And seriously, I love this paper when it came out. It's one of your best papers, Seb. Really, really nice. But he made these, beautiful idea, he made these very short symmetrical diblock polymers of lysine and glutamic acid. At neutral pH, they're soluble. If you lower the pH, the glutamic acid protonates and becomes the hydrophobic domain. And if you go to higher pH, the lysine becomes the hydrophobic domain. You have these invertible vesicles. And completely reversible. Really, really cool work. So now we were thinking, could we do this, but could we do it here at neutral pH? So we sought out to make these molecules. This turned out to be a little more challenging. If I jump back a second, when we make these polymers, we want to oxidize these sulfurs, but we want to alkylate the ones that were here. it's hard to alkylate one part of a polymer and oxidize the other part of the polymer. So what we had to do was we had to first make the polymethionine sulfoxide segment. So we polymerize methionine NCA, then we oxidize it and we purify this polymer and use it as a macroinitiator, already as the sulfoxide. Then we add our tert-butyl carboxymethyl cysteine. We cap the chains to get rid of that enamine. We use TFA to deprotect, and we get here. So we have a polymethionine sulfoxide here, and we have a sulfur thioether here. The next step is alkylation. The alkylation only occurs at the sulfide. It does not occur at the sulfoxide. So we can essentially have one part oxidized, one part alkylated. Once we alkylate here, we get these polymet sulfoxide dihydroalanine block copolymers. I would argue that these copolymers are made up of natural amino acids. These are both occur naturally. We can make these with a variety of different segment lengths. We initially chose ones with about a 60 mer of methionine sulfoxide because with previous vesicles we found like about a 60 mer of met sulfoxide and a 20 mer of, say, like leucine were great vesicle formers. But we really weren't sure what to think of this, so we made a range of different hydrophobic lengths. And then we also made one that was quite symmetrical, ideally for some type of inversion properties. And these reactions all work pretty well. Casey is just a workhorse. He cranked these all out. So what happens? Well, we essentially nanoprecipitated all of these guys. We dissolve them in DMSO at a small amount of water and then just dialyze against pure water. Remarkably, at least in my view, everything makes vesicles. They all make sub-micron vesicles. Even ones with very long hydrophobic segments make sub-micron vesicles. We don't really see anything else. If we were using alpha helical or beta sheet hydrophobic segments, this would not occur. We would start seeing fibrils and sheets and all sorts of other things. The way we image most of these, and we have lots of, these are just single vesicles, but we have low mag views in the publication. We add some dioctide to essentially stain the membrane, hydrophobic dye. We make these in the presence of Texas red dextran. We dialyze away everything that's not encapsulated, and we can see these are vesicles. So we don't do nearly as good a job as Seb's lab can do in terms of vesicle characterization, but we're pretty sure these are vesicles unilamellar. And so really a broad range of lengths will work for this. But for further studies, we chose to work with a symmetric one to see if we could invert it. So the first thing we looked is how well do these respond to some stimuli. And so what we did was we looked at these under essentially like cytosol mimetic conditions. We used 10 millimolar glutathione as our thiol. We do this in pH 7.4, 37C physiological salt. This is our suspension of vesicles as we form it. This is the cartoon version down here. We have encapsulated Texas red dextran. What we do is we just wait over time, and then at certain time points, these samples are dialyzed. So anything that's released gets pulled out. What we can see is if we have glutathione, we initially have a lot of Texas dextran loaded. After 13 days, there's really nothing left, and this is quantified down here. So if you add glutathione, we release the contents. If you do not add glutathione, you retain a decent amount of the content. So this essentially seems to work. We start with this polymer. Glutathione reacts with it. The glutathione will attach to the dihydroalanine. This will get reduced. We do isolate the polymer here and confirm that all of the dihydroalanine was modified and about 90% of the met sulfoxide was reduced. So this potentially is a good way to release cargoes inside of cells. But what about vesicle inversion? Well, this doesn't work at all here. The problem is glutathione is really not the best choice of thiol. When you make this polymer that has glutathione attached to this dihydroalanine, that polymer is really not very water soluble. And so you go from vesicles to crud. So that didn't work well, but it was an easy problem to solve. All we need is a thiol that forms a water soluble polymer. And we already knew what this was. We can use mercaptoacetic acid. So now we do this, essentially the same reaction. We start with these vesicles where the hydrophobic domain is dihydroalanine. We mix them with mercaptoacetic acid. The thiol adds to the dihydroalanine to make carboxymethyl cysteine, which we know before is that's what we started with. That's water soluble. And we reduce this to the sulfide. And this is the product. Vesicles. They look almost the same. They're similar size. We did analyze these samples and they are completely modified. They're completely modified to this. So what happens in this reaction mixture, the vesicles react and then they reform as inverted vesicles. There's a little bit of Texas red that it remains encapsulated. I think that's just material that's present in the media before we dialyze the non-encapsulated part out. So it actually does work. You can just treat these with thiol around neutral pH, and you go from these vesicles to those vesicles. So it took us 20 years, Seb, but we finally did it. So we're quite interested in this. We're also quite interested in seeing what else we can go in this direction. This was really just a model system, but it looks like it actually works quite well. I'll finish with one last little bit. And this was an observation we had with the ones with the shortest hydrophobic lengths. When we do nanoprecipitation with these, most of the time we get these tubular vesicles. And so this was weird. All of their competitions, we only see spheres, but these ones we get tubes and they're a couple microns long. So we were scratching our heads a little bit, but then of course we remembered Jan van Hest has done a tremendous amount of work in this area of transforming vesicle shapes. Especially, I mean, under different stimuli, if you put these under osmotic stress, and you have a semi-permeable membrane, or if you add crosslinkers, you can change these in different ways. Jan has published tons of stuff on stomatocytes and also prolates. These form tubular vesicles. That was really quite interesting. So we said, okay, we're definitely going to look into this. And it works. So if we take this met sulfoxide 60, dehydroalanine 20, and we do our normal nanoprecipitation, but instead of dialyzing against water, we dialyze against water with sugar. And the idea is that the water can pass through the membrane, but the sugar cannot. We always get tubes. We can also run it backwards if we have tubes, and then we plasticize the membrane by dialyzing against DMSO water, and let that go for a while, then we essentially quench it with water, we go back to spheres. So it looks like it's exactly what John was saying. You can essentially use osmotic pressure to change these. And we can also do this with all of the samples. Maybe not, we haven't tried all of them, but we take the ones that would only make spheres before, dialyze in the presence of glucose, and now all we get are tubular vesicles. So it looks like with we really never could do this with ones that had alpha helical hydrophobic segments. But the dehydroalanine segments seem to have a good amount of flexibility in them and also a good amount of permeability to allow this to happen. But we can also control the shape of these quite easily. Why we get this prolate extension as opposed to sort of stomatocytes, I don't know. Jan may have some ideas there. But it's important to remember as well, these are also still quite reactive. These will react with thiol, so these shapes will also invert amphiphilicity if you throw thiol in. Will they actually invert into a tubule? I really don't think so, but there is a lot of potential here. So this is just results from maybe 2 or 3 weeks ago, but I thought they'd be quite interesting for this meeting. So with that, I'll finish up. I have to thank Casey, who did most of the new work that I showed you today, and also, again, Seb for being a great friend and collaborator for all these years, and happy to answer any questions.